of nucleic acids, starting from the beginning from the monomers of nucleic acids, as the nucleotides. And we went ahead to look at how these nucleotides bond to give a nucleic acid chain with a phosphodiester bond between the nucleotides. And for a double-stranded to give you the double helix structure. And then in the last lecture, we looked at the function of the nucleic acids, looking at processes of replication, transcription, translation, and we focused on the processes of transcription and translation as gene expression. That is transcription of genetic code from DNA onto a messenger RNA, and the messenger RNA is then translated into a protein, and that protein can become an enzyme, a hormone, a neurotransmitter, and that's what is going to result in the regulation of a biochemical response. So we have dealt with gene expression previously. Now we are going ahead today to take a look at regulation of the genes. So we can dive in and over the course of maybe 1 hour, 20, 30 minutes, I should finish, and then I will open the floor for you to make comments, ask questions, anything that is not clear, or if you have some contributions to make, you can also make those contributions. The chat is opened. I'm monitoring it. So if you send anything there, I'll see it. Alternatively, after I finish, when you note your questions or concerns down, you can put up your hand and I will enable your microphone. Call your name and then you can speak. So let's start with the types of gene regulation. We have two types of gene regulation, and these are positive regulation and negative regulation. And positive regulation results into the increase of gene expression. And that's going to be by some regulatory element, which we'll refer to as a positive regulator. And negative regulation will be the opposite of positive regulation. And that is going to be decrease in gene expression. And the regulatory element that will be responsible for that negative regulation, we refer to it as negative regulator. So these two are simple and understood, the types of gene regulation that we have. Then let's go ahead to also categorize the genes, the types of genes that we have. Here too, we have two types of genes. The first one we refer to as constitutive genes. They are also sometimes referred to as housekeeping genes. And that's because the product of these genes, and remember, when we say product of genes, we are referring to proteins, because we already know that when genes are expressed, what we get from them are proteins. So the proteins are required all the time in the cell. So these genes are constantly being expressed. They are not under any regulation. So for instance, you talk of the process of glycolysis and Krebs cycle. These processes are occurring in the cell. These are processes of metabolism, metabolic processes occurring in the cell. And they are constantly occurring because these processes are required to generate energy to the cell. So the enzymes that catalyze those glycolytic reactions and the reactions in the Krebs cycle, those enzymes must constantly be produced. So their genes are constantly being expressed. So those proteins are not under any regulation. If at any point in time, those genes that code for those proteins that become the enzymes in those reactions should stop working, it means there will be no energy for the cell. And so they are referred to as housekeeping genes. They must keep the house before any other thing will be happening. So those are constitutive genes. The second are referred to as inducible genes. And as the name suggests, inducible genes can get their expression increased by an inducer. And obviously that inducer usually is going to be some substrates that will be inducing the process of the gene expression. And you have what we refer to as a repressor that can decrease the gene expression. So obviously, as we continue to talk about gene regulation, it means that the genes that we are going to talk about in terms of gene regulation are inducible genes. They are not constitutive genes, because we have just known that the constitutive genes, also known as housekeeping genes, will constantly be expressed, and they are not under any regulation. Now that we have cleared our minds on these simple terms, we can now dive in to look at gene regulation. We'll take it into two major parts. We'll first look at gene regulation in prokaryotes briefly, and then we go on to look at gene regulation in eukaryotes. Now, in prokaryotes, the very first concept about how genes are regulated in prokaryotes was proposed by these three gentlemen: Francois, Jacob, André, Michel Wolff, and Jacques Monod. All of them are French scientists. I think Francois was a biologist, André was a microbiologist, and Jacques Monod was a biochemist. So they worked together. And they first proposed the concept of the operon in bacteria, particularly referring to E. coli, because E. coli was the first bacteria in which the operon concept was demonstrated. And of course, they won some Nobel Prize in Physiology in Medicine in 1965 for their work on the operon concept. Now, what is the operon concept? So, the operon concept, just refers to the fact that, you have bacteria, nucleic acid in the form of plasmid. So it's a circular structure. And that plasmid, that circular structure, is having portions of it that are coding for different proteins. Those portions are what we refer to as genes. So like you have a circular structure, a nucleic acid, and as you move along that circular structure, the plasmid, you have fragments of the plasmid coding for specific proteins. So some proteins are what is referred to as operons. Now, the first operon that was actually demonstrated in E. coli is referred to as the Lac Operon because it was involved in the metabolism of milk sugar lactose by E. coli. And that was introduced in 1961. Now, we can move on to look at the operon, the complete operon structure. How does it look like? So, if you take the fragment of the Lac Operon, you can call it the lactose operon. That Lac Operon is having specific fragments in it. So the whole structure of the Lac Operon is having some specific fragments in it coding for different proteins and also serving different purposes. So for instance, the structure of the Lac Operon is having some regulatory region where you can see is labeled I and is red. And that I is for inhibition. So that is for the control or the regulation of the gene. Then you have the promoter site. And it is actually that promoter site, P, that RNA polymerase will bind to for the process of transcription. Because mind you, this is DNA, this is a DNA strand we are talking about. Then you have the operator region, which is responsible for the repressor. So the repressor that will come to bind onto gene expression, the expression of this whole gene. If a repressor comes to bind onto that operator region, the gene is not going to get expressed. Now, we see regions labeled Z, Y, A are referred to as structural genes. And the reason that is because actually When this gene is expressed, what we get from it is from these particular regions, Z, Y, A. The Z region will give us a protein, beta galactosidase. That is an enzyme. The region Y, when expressed, so that region Y is coding for galactoside permease. That is also an enzyme. And the region A is coding for another enzyme, galactoside acetylase. Now, how does this gene, operate? Now we've seen the structure. Like if you take that gene, it contains these specific portions. So now let's move on to take. So first of all, the first region, I, which is responsible for inhibition. That region, when transcripted into a messenger RNA, see that it is a tetramer, meaning that you are having 4 subunits of proteins that come together to give you that structure of the protein, which is the inhibitor. And that inhibitor, that is the repressor. And that repressor is what will go to bind to the operator region. And once it binds onto the operator region, it represses the gene, meaning the gene is not going to get expressed. What it means is that the gene itself is having an element of regulation of itself, because it is within the gene that you have the inhibitor region that is getting transcribed into a messenger RNA and producing a protein, which is a repressor that comes to bind onto the operator region to inhibit the gene. Because the promoter region we've seen is responsible for, that is where you have the RNA polymerase coming to bind for the transcription of the gene. And then you process of translation. So what it means is that this regulation is actually at the level of transcription. What that means is that the repressor is controlling the process of transcription. So once it inhibits the process of transcription, it means the gene will not get transcribed in the first place for you to have a messenger RNA. That will then go further into the process of translation to produce the proteins from the regions Z, Y, A, those structural proteins. The beta galactosidase, the galactoside permease, and the galactoside acetylase. They will not get produced. Now, in this case, with what we looked at from the beginning, it means the repressor is what? A negative regulator. And it also means this type of gene regulation is negative regulation. That's what it means. Now, another thing that we can talk about is the fact that, you see, you are referring to a fragment of a gene. So a gene that is coding for 3 different proteins. And any gene that is coding for, different products is referred to as polycistronic. So a cistron is a unit of a nucleic acid that is coding for one protein. But here you see that the gene is coding for more than one protein. It's coding for 3 different proteins: galactosidase, permease, and acetylase. And because of that, it's referred to as a polycistronic gene. And the messenger RNA that is being produced from it is a polycistronic messenger RNA. Now let's take a look at how, the mechanism of action of the inducer. And the inducer will be what? The inducer is lactose. The inducer is lactose. So lactose is the substrate upon which the bacteria should be acting. It's a milk sugar that E. coli should be breaking down to get energy from. Now, you have the inhibitor region that is getting transcribed into a messenger RNA. And from the messenger RNA, you have translation to have the repressor, the tetramer. And we've seen that this tetramer, this inhibitor or the repressor, goes to bind to the operator region to repress the expression of the gene at the level of transcription. That is preventing the process of transcription. But in the presence of lactose, which is a substrate that the bacteria should be breaking down, that lactose goes to bind onto the repressor. And once it binds onto the repressor, it makes the repressor inactive. So the repressor will not bind to the operator region. And once the repressor is not bound to the operator region, you now can have the RNA polymerase, together with some catalytic activator protein, which forms a complex with cyclic adenosine monophosphates coming to bind onto the promoter region. That will be the initiation complex. That is initiation of the process of transcription of the gene. So once you have this RNA polymerase and this energy complex bound to the promoter region, you can have now the process of transcription, and that produces the polycistronic messenger RNA, which then will undergo the process of translation to produce the proteins: galactosidase, permease, and acetylase. Now, what are the functions of these proteins? Now, galactosidase, you can see even from the name that galactosidase will be responsible for breaking down the lactose molecule into glucose and galactose. And so, because lactose is a disaccharide, disaccharide consisting of glucose and galactose. So galactosidase is responsible for breaking the glycosidic bond between glucose bacteria can now make use of those monomers, those monosaccharides for energy. Now about permease. So once the process of the expression of the Lac Operon starts, that is when there is the presence of lactose, when lactose enters into the cell. And this process of gene expression of the Lac Operon starts, the permease goes to bind onto the cell membrane of the bacteria, making the cell membrane more and more permeable for more lactose molecules to enter into the cell until all the lactose molecules are consumed. And once all the lactose molecules are consumed, it means now you are going to have less concentration of lactose. And so these lactose molecules that are bound to the repressor will then dissociate away from the repressor, and the repressor goes to bind onto the operator region. That will result into the end of the process, because then it will be switching off the gene. The gene expression will stop at that moment. How about acetylase? Acetylase, as the name suggests, should be involved in some acetylation process. That is addition of an acetyl group. It is actually not clear really what this enzyme does, what is its purpose. But these two, galactosidase and permease, they are clear. Now, we refer to molecules that are used the Lac Operon as gratuitous inducers. These are their structure looks very much like lactose, but it's not lactose. So they are structural analogs of the inducer. So usually in the lab, mostly I think they have something, is it IPTG, some isopropyl thiogalactoside or so. That is what is used to, sort of induce the gene, and then it can be steady. So it's sort of a switch on the gene to see its activity. Because it's not lactose, obviously there's not going to be a continuation of the process into the production of this polycistronic messenger RNA that will result into the production of the enzymes to break down the inducer. If it was the inducer itself, the proteins will be produced, and if the proteins are produced, they will consume the inducer and the process will end. But because you are having the inducer as just a structural analog and not an inducer itself, it will switch on the gene, but it will not get consumed because the enzymes will not be produced. So the gene can be on for some time and then it can be steadied. Right. Now, I think we saw the process when I referred to the fact that you are having a catalytic, activator protein complex with a cyclic adenosine monophosphate, which is a positive regulator in that sense. Now let's take a look at this much more. So we have lactose, which is the inducer. Remember, lactose is a sugar, but it is not the primary sugar that is used for energy. The primary sugar which is used for energy is glucose. So in the presence of glucose, it means lactose will not be consumed by the bacteria. In all of these, you think of it this way, like you buy milk and you open the milk. If you put the milk down, say that the milk gets spoiled. If that milk is contaminated with E. coli, the bacteria, because the bacteria is going to feed on the milk sugar, the lactose. I think if you think of it that way, it will be much, much clearer practically in your mind. Now, say in terms of carbohydrates that provide energy to the cell. E. coli, the cell, will preferably go for glucose than to be going for lactose, because the glucose is the primary source. So, in the presence of glucose, the Lac Operon is inhibited. And how does that work? So if we are looking at a diagram on the right-hand side, if you have glucose that is entering into the cell. So we have this line referring to the cell membrane. So this is a cell membrane. And it is inside of the cell. So the glucose is from outside of the cell entering into the cell. So it passes through the membrane and gets into the cell. Now that glucose inhibits the enzyme adenylyl cyclase, which is responsible for the conversion of ATP to cyclic adenosine monophosphate. And mind you, you are having a catalytic activator protein, which forms a complex with cyclic adenosine monophosphate, which acts as a positive regulator. You see it here on the promoter region. You have a triangle with a small red circle on it, just to show the complex, where you have the triangle referring to the catalytic activator protein and the red circle referring to the cyclic adenosine monophosphate. The two of them bind to give you the complex that binds to the promoter region, activates the Lac Operon for RNA polymerase to come and bind onto that also promoter region for the process of transcription to proceed. Now, in the presence of glucose, glucose comes to inhibit that enzyme, which is responsible for converting ATP to cyclic adenosine monophosphate. What it means is that once you don't have cyclic adenosine monophosphate being produced, you cannot have this complex formed to come and bind onto the promoter to activate the gene before RNA polymerase comes in, and then the process continues. So the process ends here once you don't have cyclic adenosine. So you can see lactose is acting as an inducer And the opposite of it, that is the molecule that will induce inhibition, is glucose. And you are also seeing that you have a repressor molecule which is being produced from the inhibitor region of the gene, which goes to inhibit by binding to the operator region. And you have the opposite of that being the complex formed between the cyclic adenosine monophosphate and the activator protein that comes to bind onto the promoter rather to cause positive regulation. So you are having both sides of what we talked about from the beginning. Positive regulation will result into expression of the gene. Negative regulation will result into inhibition of expression of the gene. And so the gene is regulated, is controlled in that manner. Now we can take a more complex operon. We're looking at the Lac Operon, which was producing 3 proteins, just 3 proteins. A galactosidase, a permease, and an acetylase. Now we're taking an operon, which is responsible for the synthesis of enzymes that are involved in the synthesis of tryptophan, an amino acid. It's an important amino acid. That's an aromatic amino acid. Of course, if you study metabolism of proteins, you come to see that amino acids will get synthesized from smaller molecules. And from one amino acid, another one is synthesized all the way. And you have these amino acids now joining to give you the proteins. Now, we saw the Lac Operon as an inducible, gene, because you have the repressor that will be on the operator region. Then, when a substrate, lactose, enters into the cell, the lactose goes to bind onto the repressor, and then the repressor leaves the operator region. Then the gene will get expressed. The tryptophan operon is a repressible gene. So what we mean by that is that the product which is to come out of this whole process of expression of the gene, which is the process of transcription and translation, is tryptophan. But that tryptophan acts as an inhibitor to one of the enzymes that are produced in the process, antranilate synthetase. That is referred to as feedback inhibition. All right, now let's start from the beginning. That we are looking at, so the top, this is the operon. So that's the structure of the gene. And we have the different fragments responsible for, coding for different proteins. And these are labeled A, B, C, D, E. I mean, reading from the right-hand side, you have A, B, C, D, E, which means This operon is coding for 5 proteins. 5 proteins. The Lac Operon was coding for 3. This one is coding for 5. But there's something more interesting about this, which is that you will think that if it's coding for 5 proteins, then you just will have the messenger RNA transcribed along it, and then along the messenger RNA, the various corresponding fragments of the messenger RNA will just code for the proteins. So they will get translated for the proteins. But actually, this operon gets transcribed into 2 types of messenger RNAs. So from it, 2 types of messenger RNAs are transcribed, not only one. One of them gets transcribed along the whole stretch of the genes. But it is only the D and the E region that are active in that messenger RNA. The second gets transcribed only from the A, B, and C regions, just those 3 regions. And it is that one that gets translated into 3 different proteins. So the messenger RNA, which gets transcribed from just the A, B, and C region, that one will have all the 3 fragments being active and cool. The messenger RNA that gets transcribed along the whole 5 regions of the genes, it is only the D and E regions that are active. Why so? We don't know. Why not just transcribe all of this one and then you just translate all the regions? But somehow, when you have the full stretch being transcribed into the messenger RNA, some portions are inactive. Now we see the different regions when they get translated, the messenger RNA, when they get translated, they produce different proteins. So we have moved from the messenger RNA level, and now we are looking at polypeptides, which means now there is a process of translation to give you some proteins. And even these proteins, so for instance, you can take these polypeptides, which is tryptophan synthetase alpha and tryptophan synthetase beta. The 2 of them actually form a complex. So you now have an enzyme complex being formed. So just like the way you have hemoglobin molecule, 2 alpha chains, 2 beta chains, the same thing you are seeing here, also a tetramer. You have a tryptophan synthetase, which is acting in a particular reaction. Now, you see, there is a reaction below, catalyzed reactions. It is actually this reaction that the gene, when it gets expressed, comes to control. And what is the end product of the reaction? Tryptophan, an amino acid. It means it is the process of synthesis of tryptophan that the gene is controlling. So that you have different proteins that are produced from that gene. And you see, you are having a reaction chain. It's not just a single reaction. So it's a reaction pathway, one reaction after the other, one reaction after the other, which leads to the final product, tryptophan. Now you see, you have the proteins, like the first enzyme, which is antranilate synthetase. This is also from the D and E region of the gene. This enzyme comes to catalyze the first two reactions. So you see, you have the beginning of the reaction, chorismate. Glutamine comes into the reaction and gives you a product, antranilate. Then you have some phosphoribosylpyrophosphate. Pyrophosphate. You are having another product, phosphoribosyl antranilate. The reaction continues, and you see the arrows are pointing onto those arrows of the reactions that are leading to the formation of tryptophan. What it means is that those enzymes, those proteins that have been synthesized from the messenger RNA are catalyzing those. The gene is regulating the process of synthesis of tryptophan. That's why it's been referred to as tryptophan operon. Now, when you look at some things like this, I know how sometimes students, the moment they see some diagram, and it's like many arrows, and to them it's looking like this is a lot of things at once. But no, you just have to take your time. I mean, this is the thing. Imagine that you find yourself in some jungle somewhere and you are given a map. On the map, everything is there. Where you are found, you have where you are. All you need to look for is the nearest safe place. And it's on the map. You can't throw the map away and just be walking about anyhow. You study the map and follow direction. That's what you do. So the same way, no matter how complex like you see something like this, a path, even this one is quite easy. It's just words. It's not like we don't have like chemical structures even over here where you have to be looking at where some part of a molecule is moving to go and join another one and things like that. No. All what you are seeing here, they are words. So what do? Follow the arrow. Have the gene like this, and then straight it will just, or maybe if you take this reaction below here, you could just do chorismate, and the arrow will just be drawn chorismate, and then the next thing you see is tryptophan. Yes, it's true that chorismate, maybe they will put glutamine there because that's the beginning of the reaction, that chorismate and glutamine react, and in the end, you will get tryptophan. But there are intermediates. Things happen in the middle before the last product, the end product is received. So yes, in the secondary school, you were studying chemical reactions like that, simplified. You take reactants and then you take products. Now you have moved a little bit. You are in the university. So you begin to look at intermediates in the middle. What is happening there? So just a little bit stretch of the brain, and you'll be feeling some resistance in your brain that no, you don't want to move away from what you already knew from the secondary school, but you have to grow. So step by step, arrow by arrow. You know, when you start to go to school from primary school, I mean, let's take it from nursery. Actually, the most important thing which you were taught at that time was to read and write. If even for you to use your 3 fingers to hold pen or pencil was a skill that you had to be taught to be able to write. So write and read. Now you are in the university, you have something in front of you. Read it. Read it and have an understanding to it. That's all. So I remember some student, I think last semester or so, some exams, I just gave some diagram and I asked you to explain the process of so and so. The thing is in front of you, it's there. Just look at it and write what you are seeing. Isn't that much easier than the thing is not in front of you, but you are being asked, explain how so and so and so, and you have to be thinking. It means how I have to uh, have it all in your head to be writing. This one is in front of you. The diagram is there, and the question is being asked, so that you just look on the diagram and say what is happening, because you should be able to read. And this is, it's professional reading. So a lot of you, yes, I know you will be very much interested when you are in the lab and you are just, mixing things and getting results, and then you just go and give result to somebody else to translate, and that is all. Now, you should be able to also look at the result and have an understanding to it, because you must grow. You cannot be there all the time. You must move on. All right. So yes, we see here too, there are some control sites like the promoter region, operator region. There's even some activator region, some what is referred to as the second control site. And then you have some other promoter region also here, which is for the A, B, and C. That is particularly for the shorter messenger RNA. So those things are also there that you can take a look at. All right, so that's the tryptophan operon. So that was more complex than the Lac Operon for the prokaryotes. So we can move on and take a look at gene regulation in eukaryotes. Gene regulation in eukaryotes. Of course, some of the things we'll see that they also occur in prokaryotes as well. The first we are looking at is gene regulation by methylation. By methylation. So you see, when we were looking at the structures of a nucleic acid with RNA. We saw primary structure. We saw secondary structure. We saw tertiary structure. Yes. We saw tertiary structure. And I think we even saw quaternary structure. Because quaternary structure will only be that you have RNA molecules with different, or you have one or more, tertiary structures in them, and they are in a complex together. But when we were doing DNA, we only talk about primary structure and secondary structure. I expected you people to ask, so why is it that DNA, we are not seeing tertiary structure, quaternary structure? Nobody asked. Everybody just kept quiet. You see, you have DNA molecules that are double-stranded, and you are having double helix as the secondary structure. But in the nucleus, those double helix structures actually wind. First of all, they form chromatins. They pack and form chromatins. And after they form the chromatins, they bind around proteins, what we refer to as histones. Sometimes they say histone proteins, but it's just a repetition. Histones, because it's known, histones are proteins. So you see, tertiary structure of DNA is going to be the chromatins. And the quaternary structure is when these chromatins bind around the histones to form what we refer to as nucleosomes. So DNA is packed in the nucleus as nucleosomes. Now, you know, from the last lecture, we were looking at the process of transcription. So you know that in order for a fragment of DNA to get transcribed, that fragment must be exposed. If the fragment is not exposed, it cannot be transcribed. It's a double strand, and you are having hydrogen bonds between the nucleotides. If it's not opened, the strand cannot get transcribed. It must be opened to be exposed in order for it to get transcribed. And here is the case, you are also having it packed like this. How does it get packed like this? So you see, you have DNA that the monomers in DNA, they are nucleotides. And the nucleotides are negatively charged. They are charged on the phosphate group. And they are being wound around proteins. And these proteins are made up of amino acids. And the amino acids exist as zeta ions, which means they are having both positive and negative charge on the carboxylic acid group because it gives out hydrogen. And on the amine group, it receives hydrogen there. It gets protonated and is carrying a positive charge. So what it means is that those amino acids can also be charged so that you are having some negative charges that can be resulting into repulsion with the negative charges on the phosphate groups of the nucleotides that are in the nucleic acid, the DNA. So in order for these nucleic acids to pack together with the proteins like this, you needed to do away with the charges. And that is done by the process of methylation. Methylation is just addition of a methyl group. So a methyl group attaches onto the histone protein. Methylation can also get attached to the nucleic acid, the phosphate groups, and then no charges, then they can bind together. When they bind together, it means in this manner, we are seeing, so there's a portion here, red, like that's a gene that is supposed to get transcribed and get translated. So it's supposed to be expressed. But that gene, when bound like this, becomes inaccessible. And so the gene is inactive. That's a form of regulating the gene. So that's by the process of methylation, making the gene inactive. It cannot get expressed in this form. If you go to look further at histones, you see here too is being depicted. You can see clearly histones are also tetramers. That's why they are depicted like that. 4. You have it, being shown as having 4 subunits, and then you are having the DNA wound around them. So you have these nucleosomes that are packed together. Right. The second form of regulation is acetylation. Acetylation. So acetylation will be doing the opposite of what methylation is doing. So when you are having acetylation of usually you have these amino acids that are positively charged. Those amino acids are having extra amine groups like lysine. So those amino acids, they can have attraction for the phosphate group. But that is the phosphate group in the nucleotides. So they will be binding. But when those side chains, those amino acids that are having those positively charged, when they get acetylated, it results into unwinding of the nucleosomes. And so now that region of the DNA, the fragment that is supposed to be expressed, is now exposed, is now accessible. The gene is now active to be transcribed and translated. So you can have gene expression. So that is the process of acetylation. Methylation, acetylation, you'll find in eukaryotes. So we can look at acetylation much more. So this is a histone with these black lines wound around the histone proteins are referring to the DNA molecules. And the histone, showing a lysine residue. And that lysine residue is having a side chain that's having an NH3 plus, the positive charge on it. And you see acetyl-CoA comes into the reaction and an acetic group. So you see acetic group, acetyl group now comes to bind onto that nitrogen of that amine group. That is acetylation. Now lysine is acetylated. So this is what I was referring to. Now you can see it clearly here that you can have such acetylation, which will result into activation of the gene that is to be expressed. Of course, that's a reversible reaction, and so you can have deacetylation. So deacetylation will do the same thing as methylation is doing. All right. Now, you see, so far we have been talking about genes. And genes are nucleic acids. But we also talk about gene regulation by peptide activators or inactivators. What that means is that there is an interaction between nucleic acids and proteins. Because right from the beginning, from Lac Operon, we saw that the inhibitor region gets transcripted into a messenger RNA. The messenger RNA gets translated into a protein. It is that protein that goes to bind to the operator region. So a protein is interacting with a nucleic acid. So both in prokaryotes and eukaryotes, you have regulation by what you can refer to as peptide activators or inactivators. Now, these can occur in different forms. So, for instance, if you have, the numbers 1, 2, 3 from the top, 1, 2, 3. These are some specific proteins that are interacting with gene 1. That's the first gene. And once they bind onto the gene, it results into activation. So they are activators of the gene. Or you can have things like 3 proteins where you have 1 and 3 interacting directly with the gene. But the third protein, which is numbered 4, only comes to link these 2 proteins that are interacting directly with the gene, and that can result into activation of the gene, which means if you don't have protein number 4, which comes to link the 1 and 3, the gene is not going to be activated. So different ways that you can have peptides interacting with nucleic acids to activate them. You see another example where you have, say, protein number 1, protein number 3. They are bound on the gene. Now there is one protein number 5. When it binds on the gene, it prevents protein number 2 from binding. It's like that is an inhibitor. It makes the gene to be inactive. Once it binds, it inhibits protein number 2 from coming to bind onto it. So if you look at it like it is like gene number 1, which we are seeing from the top, the same diagram that we are looking at, you can say where protein number 5 is binding is sort of some allosteric site, just the same way that enzymes behave. And another protein number 5 binds there, and it prevents number 2 from coming to bind. And that renders the gene inactive. So there can be so many complexes, complex systems by which proteins will activate or inactivate genes. Right. So that's to look at it broadly, that you can have different ways. Now, you see, when you talk about enzymes, which are proteins, enzymes act on substrates. They act on some specific compounds. That is to increase the rate of their reaction. And if you studied enzymes, the enzyme is a big protein, but it's not that any portion of it at all is having interaction with its substrate. You have some specific portion of the enzyme, the protein, that will have interaction with the substrate. That specific portion we refer to as the active site or the active center of the enzyme. The same way, now you have nucleic acid that is interacting with proteins. It is not any part of the protein at all, which is just, having interaction with the nucleic acid. You have some specific portion of the proteins that have high affinity for specific portions of the nucleic acid. Usually it is going to be, see, when you look at the secondary structure, when we look at the double helix, we saw the major groove and the minor groove. And I mentioned that those major and minor grooves are the places where you can have interactions with proteins. And usually these proteins are what we refer to as transcription factors. So you have specific portions of the proteins that have, some form of hydrogen bonding and van der Waals forces between some specific base pairs, like maybe 5 base pairs within the major groove. They have such an interaction with these nucleotides in the grooves of the nucleic acids. Those portions are what we refer to as protein motifs. So just like we have active center for the enzyme, for this particular interaction with the nucleic acids, we have protein motifs. We have 4 major types of these protein motifs. We have what we refer to as helix-10 helix, zinc finger, Leucine zipper, and helix loop helix. Not complex. It's just about looking at the structure, how it looks like, and the name is given to it, just the way it looks. So for instance, helix-10 helix motif. It's looking at so if you have, the black is referring to a DNA fragment, and then the red on it is the protein motif. And you can see there is some alpha helix structure, and there is one loop. After that loop, you have another helix structure. So it's referred to as helix-10 helix. Actually, the protein, the repressor, the inhibitor, which is produced from the Lac Operon, which goes to bind to the operator region of the Lac Operon, has this helix-10 helix motif. Similarly, you have the interaction of, the complex of the cyclic adenosine monophosphate with an activator protein. That's a protein which is having interaction with the nucleic acid. There too, the motif is a helix 10 motif. And usually it's just made of, that's like an oligopeptide. That consisting of about just 20 amino acids and is interacting with a nucleic acid. The zinc finger motif. So you see, it looks like a finger. If you look at the top, you see, looks like a finger. Like if you can put your thumb on it, and you see, it looks like a finger. Now, the interesting here is you have the zinc ion at the center of this protein, and around it you are having some cysteine, amino acid cysteine at different positions in the protein or the peptide chain. And they find themselves at specific positions such that you are having some disulfide bridges. You remember, cysteine is one of the amino acids with sulfur in it. And you also remember that when we're looking at the tertiary structure of proteins, one of the bonds in there was disulfide bridges. So here you are seeing, what you see here, C, and then there is an arc red, CC, arc red around it. Those are disulfide bridges. And you also have the same thing. So you can have on both sides, cysteine, two of them having that disulfide bridge between them. Or you can have a zinc finger that is having cysteine on one side, having a disulfide bridge between them. But on the other side, you are having a histidine. And that histidine can be having some form of hydrogen bonding between them over there. Now, these two you find, so here it's clear that you find it binding to the major groove. So it's here. And the contact they have is with like 5 base pairs in the groove. And you find this sort of interaction with some steroid hormone receptors. I mean, when we come to hormones, when we go ahead, we'll come to study some fundamentals of endocrinology. And you realize that there are some hormone receptors, which are steroid hormone receptors, that penetrate through the cell. Because steroid is a lipid, so it can pass through the phospholipid bilayer of a cell into the cell and even go to through the nuclear membrane and goes to interact with the nuclear material to control gene expression. And there, the interaction that it has with the nuclear material is with the zinc finger. You also have what I refer to as transcription factors. They are proteins. I think the animations I sent to you that you were watching about transcription and translation, they refer to some transcription factors. They are proteins. And those transcription factors are having interaction with the nucleic acids. So some of them have the zinc finger motif. These are the specific portions of these proteins that interact with the nucleic acid. You have leucine zipper motif. So this one too, just looking like a zip, and it's called leucine zipper because this is rich in the amino acid leucine. You have some repeated sequences of leucine. And you see it forms some dimeric structure. So two, you know, parts of this protein structure that look alike, like having this leucine rich chains, and you see it having interaction with the nucleic acid. Also find it in transcription factors, like what is referred to as the force and John, which you see in processes of cell proliferation and the growth of cells. You find a leucine zipper having such interaction with nucleic acid. I think this should be the last, like the helix loop helix. So here too, you can see the helix structure of the protein, helix, which are having interaction with the nucleic acid. And you see a loop on both sides. And after the loop, you see another helix again. So it's being referred to as the helix loop helix. Also dimeric, because you have two subunits that are looking the same. They look similar, and they are having such an interaction with the nucleic acid. All right. Now, we can also talk about another form of gene regulation in eukaryotes we refer to as gene amplification. Now, here, the gene amplification is different. In this context, it is different from what you refer to when you are talking about, say, PCR, polymerase chain reaction. Because there too, when you are having a multiplication of the genes, we refer to it as a gene amplification. But here, in this context, it means a different thing. That is to say that you are having the expression of the gene increased in a number of times, like several folds. And that occurs sometimes because of, when there is the need for an increased rate of growth of cells. So you see this occurring in like the developmental stages of eukaryotic organisms. When, like say, the fetus, and there is supposed to be a fast production, that proliferation of cells, organs growing and all of that, you have such gene amplification. You can also have such in things like drug resistance. So for instance, you have what you refer to as methotrexate drug resistance. That's a drug which is used in chemotherapy for dealing with cancer. And that drug, after the cancer cells are exposed to it over a long period of time, these cancer cells begin to develop some adaptation to the drug, such that they begin to proliferate the more. They express their genes that are responsible for the synthesis of the protein dihydrofolate reductase. What it means is that, dihydrofolate reductase is involved in the in a metabolic process where you have what you refer to as folic acid. Folic acid is involved in cell proliferation. So you have that gene, which is responsible for coding for this protein, this enzyme, dihydrofolate reductase, being produced the more. So you have more synthesis of folic acid, dihydrofolate reductase, and then you begin to have more and more and more of the cancer cells being produced in a way to compensate for the cells that are dying under the action of the drug. All right, I see Bright has written something. I'm coming. I'll come back to you. I'll respond to you. Then you also have, for instance, gene rearrangement or gene recombination. So, for instance, in the synthesis of immunoglobulins in the immune system. So we have an original DNA that is actually coding for that protein, the immunoglobulin. There is some rearrangement, such that some portions of specific portions of the gene get rearranged, such that in the end you have what is referred to as the light chain of the protein. So it's not the whole gene which is just getting transcribed and then it goes to give you messenger RNA, which corresponds directly to the gene, and then you have the protein from it. But you have this recombination of the genes. It's a form of regulation of the genes. You can also have, I think when you go and look at some of those videos, you will see somewhere capping and poly A tail being attached in processing. So similarly, you have in messenger RNA some protective capping to the 5' end, and then the 3' end, you have some poly A tail also, protecting the messenger RNA from degradation, because degradation of the messenger RNA, you remember we mentioned that you have some catalytic RNAs, ribozymes, that are involved in the recycling or the breakdown of messenger RNA. But this sort of capping actually, protects the messenger RNA from degradation. It's also a form of regulation of the gene. Because if the gene is degraded, it means it will no longer be there, to be expressed. The messenger RNA is not there. There will not be any process of translation to give you the proteins. All right. Then the last thing we can talk about, quickly, is you can refer to, these things we've talked about like drugs that can induce the gene or result into inhibition, or some environmental chemicals, or even development of cells. We refer to things like the cell proliferation when we refer to gene amplification, because you are having in eukaryotic cells at the developmental stages, growth increases, or even in like aging, for instance, or food diet, which can also have some effect on regulation of the gene, or have effect on gene expression. These things are external to the gene itself. It's not like the gene is being manipulated, that there is some change, like we saw with recombination or rearrangement of the genes. That one is within the gene itself. But any factor which is outside of the gene which is having an effect on its expression, we can refer to as a form of epigenetic regulation. So you can see repeated what is being shown here, process of methylation, acetylation that we saw. Those processes are actually not causing any change to the gene itself. Is rather just either closing it or opening it. That's all. But they are external factors that are resulting into that. So all of these can be referred to as epigenetic regulation. All right, so now let me see. Bright Natasaki says the tryptophan operon on the long messenger RNA, there might be inactive sites. Please, on the shorter one, can't same occur? If it does, what happens then? So on the shorter messenger RNA, all of them are active. So you have all the sequences or the codes which are there on the messenger RNA, the fragments. They are all active coding for those proteins. If any of these should become inactive, it will mean that you are not going to have the synthesis of the specific proteins, which will become the enzymes. So for instance, if you say the alpha, tryptophan synthesis, that is to say that the first section, tryptophan A, that section does not produce the alpha chain. It means you cannot have the alpha chain to have this full tryptophan synthesis. So you will not be having the synthesis of tryptophan in the first place. That will mean that the whole gene is inactive. That's what it means. And Samuel says, how does exposure to nuclear reaction cause gene mutation as an external factor? I think that's what you are referring to. So exposure to nuclear reactor, that is to say, let me not say it's a nuclear reaction. Let me say it's a radioactivity. You are exposed to the radiation. That's what you want to say. You are exposed to the radiation. So radiation